It is my honor to introduce Dr. Isabel Muzzio, who is a professor in the Department of Neuroscience, Developmental and Regenerative Biology at UTSA. Dr. Muzzio obtained her PhD at Rutgers University, working under the supervision of Louis Matzel, studying plasticity in the photoreceptors of, I cannot say this name.
Vermicenda.
Crassicornis, which is a sea snail. She has conducted postdoctoral work at Columbia University, working under the supervision of the famous Dr. Eric Kandel, where she was studying plastic changes associated with learning and attention in mice. During this time, she also learned to conduct in vivo recordings in freely moving animals in collaboration with Dr. Busaki. She's moved to UTSA 6 years ago and is currently a professor and performing some amazing research on neural representations and circuits that we are going to learn today. She has also received several awards, including an NSF Career Award and nominations for the UTSA Presidential Research and Teaching Awards. If you're not speaking, can you please mute yourself?
That was my dog.
Oh, sorry, that was yours.
No. This is my dog. I was in the previous one. I apologize. He has something to say.
He's excited about the talk. So she has served in multiple national and international panels and is currently a member of the SFN Neuroscience Scholar Program. And her research is funded by several NSF and NIH grants. It's an honor to have her here today. And the floor is yours, Isabel.
Thank you so much for the kind introduction and for the opportunity to share my research with all of you. I am not an artificial intelligence person, but I hope that some of the research conducted in my lab can inspire collaborations and questions from all of you. My lab is interested, I'm trying to move here, but it's not working.
Oh. Do you want to stop sharing and try sharing?
Yes, let me, oh, okay. Now it moved. Let me see, now it's moving. Okay. It needed a few seconds. So my lab is interested in several questions. Today I'm going to be talking about reorientation, spatial reorientation, but I want you to know that for many years I have been interested in learning and memory. And we are studying how emotions influence neural representations of episodic memory. We have an interest, long-lasting interest in aging and how aging affects sleep patterns, cognitive abilities, and representations in the brain. And we also are interested in understanding how learning influences our ability to regain our bearings in space, which is known as a spatial reorientation. And this topic has a lot of implications, not just for health, but for the field of robotics. And in terms of health, I would like to point out that disorientation is one of the signatures of neurodegenerative diseases such as Alzheimer's. It also increases as we age. Is present in some genetic conditions such as Williams syndrome. And probably you all heard that one of the cognitive side effects of the COVID pandemic, the COVID infection, is to experience temporal disorientation, which people have called the COVID fog. So it is really, really important to understand, how the brain controls our ability to regain our bearings. So I will give some background because I know this audience is broad and some of you are not working with the paradigms or are not familiar with what I do. So when you are navigating in a space, there are two sets of cues that you use in order to keep track of your bearings. You use what are known as idiotetic cues, which are represented by your internal self-motion cues that originate from vestibular organ, proprioception, optic flow. And then you use all the allothetic cues, which are all the external cues that surround you. When you are oriented in a space, these two sets of cues provide convergent information that allows you to know where you are. But if you get lost in a context that you are not very familiar with, or you take the metro and you don't keep track of your start position, and you have to reorient in space, then your internal self of direction is not going to be reliable. Under those circumstances, you have to reorient only using external cues. Now, around us, there are many external cues. There are local cues, such as the position of this booth, more distal cues, such as this building, the position of the sun, the layout that surrounds you. However, across the species, it has been shown that when subjects are disoriented, there is one cue that has more importance to allow subjects to reorient than others. And this started in the 80s with the work of a psychologist called Ken Cheng. And what Cheng did was to disorient rats by spinning them around and then placing the rat in a rectangular environment that in the original study contained many cues on the walls. For the sake of simplicity, I only have one. And the task consisted in having the disoriented rats finding a reward that was hidden in 1 out of 4 cups. And what Cheng found was that the rats could not solve the task because the animals will dig in the correct corner and equally often in the geometric opposite corner, and in these 2 locations more than in all the others. So what these results were telling Cheng was that the animals were not using the non-geometric cues on the walls, even though they had directional information, because you can imagine if the rat were using this stripe wall, the rat that was not sure where the reward is would dig sometimes in the correct location, maybe sometimes in this location near the cue, but not in the geometric opposite corner. So what these results indicate is that the rats are using the shape of the layout, ignoring all the cues around the walls. So if in one trial, the rat happens to enter the environment facing north, it will say, okay, the correct corner is where I have the long wall on my left. But if in another trial, the rat happens to enter the environment facing south, it will confuse the corner that shares geometric properties. So these results have been replicated in chickens, in fish, in bees, in human toddlers, and we replicated them in mice. And it has been suggested that the geometry plays such a fundamental role in disoriented animals because it's a more stable cue. If you are lost in a neighborhood and you start paying attention to, for example, political signs to get to your goal location, those signs may be there before elections, but not after. However, if you pay attention to the shape of the layout, that cue tends to be more constant over time. So these results were replicated across a species and confirmed many times, but the theories behind explaining these results have been hotly debated for more than 30 years until we started working on this topic. One camp of researchers suggested that the disoriented subjects can show this pattern of behavior when they are trying to reorient because the brain forms an encapsulated geometric module, a representation of the layout that is impermeant to non-geometric cues. Now, this theory implicates that the medial temporal lobe of the brain, which is important for navigation, has to play a role. Now, other researchers said that the brain could solve these problems using a view matching view. This theory suggests that the animals don't have to have a representation of the layout. The animal sees the goal and matches that goal with a stored representation of some aspect of the goal. For example, long wall to my right, and then use a stimulus response association to get there. So this view matching theory says that you don't really need to have a map of the environment, only some aspect of the environment that is stored, and you match that with the goal location. So it's clear that this theory doesn't require the medial temporal lobe. So we thought that when I became interested in this topic is that if we record from the hippocampus, which is a structure that is thought to create a cognitive map, then we could elucidate behind these theories, what was right. So I will tell you a little bit about the techniques recorded in used in my lab, and also a little bit about the properties of cells in the hippocampus for those of you who are not familiar with navigation. In the hippocampus, the principal cells there show this pattern of activity in which if you record neuronal activity as the animals are moving, you see that cells have a tendency to fire or have activity in some locations and not others. This led John O'Keefe, who discovered these cells, to call these cells. So here I am showing you a video of an animal that the speed of the animal has been increased 10 times. The path is indicated in gray, and each red dot indicates an action potential. So in this example that I am showing, you can see that this cell has a tendency to fire in the lower left corner of the environment, which is what we call the place field of that cell. So we record the position of the animal over time, as well as the neuronal activity. And once we have several minutes of recordings, we generate these rate maps, which are two color dimensional representations of the recording environment that have been color coded representing neuronal activity. And in the maps that I am going to show you today, red indicates that the cell has fired a lot, and blue indicates that the animal has been there and the cell has not fired or fired minimally. Now, in addition of recording with tetrodes, my lab has implemented the miniscope technique. For the last 2 years, we also have been using this technique, which allows us to record from hundreds and hundreds of cells at a time. So what we do is to inject a calcium indicator under a particular promoter that allows us to express that indicator in a subpopulation of cells of interest. And then what we do is to record these calcium transients, which are indirect correlates of neuronal activity, at the same time that we record the position of the animal in space. And we can correlate the neuronal activity with the position to do the same types of analysis that we do with tetrodes. Now, the two techniques are complementary because the calcium imaging allows us to record from hundreds and hundreds of cells at a time. So if we are interested in asking questions about how different subpopulations of cells behave, this technique has more power. But if we are interested in questions that relate to temporal specificity, for example, firing rates or how if there is synchronization in a particular brain region with another or so on, then tetrodes provide us with that temporal specificity that the calcium imaging technique doesn't have. So in the last 2 years, we have been using both techniques to record from the brain. Now, as I mentioned, for us, it's very important to track position and head direction. In the past, we used to use LEDs to calculate the position and head direction. More recently, we have been using DeepLabCat, which is a machine learning algorithm. And the way this works is that for us, the important part, as I said, is the head direction and position. So we put dots on each side of the ear and the nose. Manually, we scored like 500 frames. Then we feed that into the algorithm. And this works amazingly well. You can see that the path is decoded very, very well. And the head is captured, the head movements are captured with very high precision. Even small movements of the head, which are indicated by the green bar, are very well recorded and captured with this technique. So nowadays we are using these instead in combination with the LEDs, which allows us to correct for any errors that can come from the LED recordings. Now, before I go into the data, I want to remind everybody about why place cells are thought to create a cognitive map of allocentric space. The place cells in the hippocampus respond to changes in environmental properties. So for example, if you record from an animal in one context with one set of cues, and then you put the animal in another context with different cues or different shapes, you will see that the same cell will fire in different locations, something called location remapping. Similarly, if you record in a room and then you change the colors of the room, the colors of the walls, what you sometimes see is that the cells continue firing in the same location, but the rate at which the cells fire is different in the two conditions. So place cells can code environmental changes through location as well as rate remapping. And they have been shown to be extremely responsive, for example, to landmarks. So the question that we had when we started recording and trying to understand reorientation is, given that place cells are extremely responsive to landmarks, if, for example, I rotate the position of this landmark, a place cell will perform a concomitant rotation. So in disoriented subjects in which it has been observed that they only use the geometry to reorient. Does it mean that the animals will ignore the landmarks and the whole hippocampal map will become aligned to geometry? That's the question that we wanted to address. And to do this, we recorded from mice in area CA1 in 3 different environments that had unique shapes. In each of the environments, we put a non-geometric cue in one of the short walls. And we trained the environments, the animals, by disorienting the mice before each trial and placing them in one of the environments, allow them to forage for 3 minutes, then remove the animal, disorient again, and start another trial. We trained the animals in each of these environments on 3 consecutive days, and the order of the environments was randomized across animals. And in between trials, we did all sorts of controls to ensure that the only cues that the animals were using to reorient were either the non-geometric cue on one of the walls or the shape of the enclosure. Now, these were the hypotheses that we had. It was possible that the non-geometric cue controls the activity of the cells. If this is the case, even if the animals are disoriented, we should see that cells are stable from trial to trial. If we found this result, this would argue against the modular theory, and it would suggest that the behavior observed by Cheng and others happens outside the hippocampus. Another possibility was that the placed cells in the hippocampus become totally erratic and unstable from trial to trial. This result would also argue against the modular theory. The third possibility was that the hippocampal cells become aligned to geometry. If that was the case, then we had different predictions for each of the environments in which we recorded. In the rectangle, we expected that because it has 2 orders of rotational symmetry, the cells will rotate 180 degrees. In the square, because the 4 angles of the square are identical and it has 4 orders of rotational symmetry, we anticipated that 0, 90, 180, and 270 should be equally represented in the rotations of the placed fields. And we, on purpose, we use an isosceles triangle that doesn't have rotational symmetry because we anticipated that in that case, even if the animals were disoriented, we should see a stability because each view of the isosceles triangle is unique. So I am going to show examples of cells recorded in each of these environments. This is a cell recorded in the rectangle. You can see that the cell doesn't rotate on every trial, but when it rotates exactly 180 degrees. This is another simultaneously recorded cell. And you can see that the population rotates coherently. This means that if from trial 1 to trial 2, there is no rotation, another simultaneously recorded cell doesn't rotate either. But if from trial 2 to 3, there is a rotation, then we see rotations in simultaneously recorded cells. In the square, as we predicted by the geometric alignment hypothesis, we saw all the angles of the square equally represented. We saw 180-degree rotations, 90-degree rotations, 0-degree rotations, 270-degree rotations. And the population rotated coherently. And as expected, in the isosceles triangle, we didn't see rotations. We only saw stability. We quantify these results using 2 methods. I'm not going to talk about this because this is published data. I won't tell you about the second experiment. I will talk about the quantifications of our unpublished data. If you have questions, I can address them at the end. So since the alignment of the hippocampal map appears to be so strong, we next ask, can we use the alignment to predict behavior? So to do this, now we train the animals in Cheng's task. So in the previous experiment, the animals were allowed to free forage in the environment. Here, what we did was to train the animals to find the reward that was hidden in one of the 4 cups. So this is a goal-oriented task. So first we replicated the results found by Cheng, showing that the animals dug in the correct as well as the geometric opposite corner more than in the other locations. We also replicated the alignment of the placed cells according to geometry, which is shown in this cell example here. And what I am showing you here is the behavior of the animal on the third day of training. So in order to predict, I want to show you something so that you notice. When the animal makes a correct choice, which I highlighted with green here, you can see that this cell fires for the most part in the upper right corner. And when the animal makes a geometric error choice, the cell seems to fire in the geometric opposite corner. So what we wanted to do is to use the alignment to see if we could predict the behavior of the animal. To do this, on day 3, we withheld 1 trial at a time. And then with the 7 remaining trials for each animal, we created an average map each time that the animal made correct responses. So for example, this is the average for cell 1, the 4 trials that the animal makes a correct choice. This is the average for cell 2 and so on. And then we created an average map each time that the animal made a geometric error. And then we asked, is the withheld trial more similar to the average correct or the average geometric error map? And to do this, we do pixel to pixel cross correlations of neuronal activity. And you can see in the example here, this withheld trial would be categorized as a correct choice map because the correlation is much higher. So using this method, we predicted all the trials for all the animals we recorded. And you can see that in all the cases, our prediction was about chance. And the overall prediction was significant, the correct prediction was significant, and the geometric error prediction was as well. So the final thing that we did was to ask, here we are using all the neuronal activity of the trial to predict the behavior. How much neuronal activity do we need in order to predict whether the animal will go to the correct or the geometric error cap? So we use the same method, but rather than using the whole trial, now we use cumulative times. So we use 1 second, 2 seconds, 3 seconds. We found that within 17 seconds, we could reliably predict about chance whether the animal was going to go to the correct or the geometric error cap. And this was super interesting to me because the median that it takes the animals to make a dig is about 33 seconds. Very few trials the animal digs in a cap before 17 seconds. This means that the alignment of the hippocampal map to geometry is so strong that we can predict what the animal is going to do just using 17 seconds of neuronal activity. And these results provided strong evidence in support of the modular theory of reorientation. And they were highlighted by one of the pioneers in the field, Randy Gallistel, because of that, because they kind of provided the strong evidence that kind of solved this debate that had been going on for 30 years. So one thing that I would like to mention that came to mind to me when we were doing these reorientation studies is that it's kind of strange that the animals are not using the features in the environment, because I have very bad reorientation abilities and I tend to use features all the time. So one thing that I noticed is that all the disorientation literature normally tested human subjects or animals in single contexts. So if I disorient each of you by spinning you around, but you know the room where you are, the only thing that you need to do in order to regain your bearings is to recover your heading direction within the context, something that we call heading retrieval. So we hypothesized that the animals use the geometry to recover heading. But similar, at the same time, in real life situations, let's say you get lost in New York, you not only have to recover your heading, you also have to determine in which part of New York you are. Are you in Midtown or are you in Washington Heights? So if you see the Empire State Building, you will say, I am in Midtown facing south. If I walk 3 blocks to the right, then I will get to the metro station. So we thought that these two processes, heading retrieval and context recognition, have to happen simultaneously to allow animals to reorient in space. So how can we test if this is the case? So at the time, one of the students, Julian, working in my lab, thought about modifying Cheng's task. And we trained the animals in 2 identical chambers that had the same shape and dimensions that only differ by the presence of a non-geometric cue in one of the short walls. In context A, we use vertical stripes, and in context B, horizontal stripes. In context A, Josh rewarded the left, and in context B, the right. So we anticipated that if the animals were using the geometry to recover heading, they will dig in these two corners in A and these two corners in B. But they will only show this dissociable pattern if simultaneously they use the non-geometric cue to say, I see the vertical stripes. That means I am in A, and the reward corner is the corner where I have the long wall to my left, and it will confuse these two corners. But if the animal saw the horizontal stripes, it will say, okay, the correct location is where I have the long wall to my right, so it will confuse these two corners. So we tested this, and indeed the mice show this dissociable pattern, which is summarized here. And these results were very important because they show that during disorientation, there are these two processes that happen simultaneously: context recognition and heading retrieval. And they are differentially sensitive to landmarks and geometry. So the last few years, I have been trying to record from the hippocampus in order to identify how these processes are represented. And the questions and papers that we have been publishing also relate to where is the geometric input coming from. So this is what I am going to tell you next. So these are limits of the modular theory, as I said, the situations of contextual ambiguity. And also we thought, if we train the animals in this task, in which the animals have to pay attention to the non-geometric cues in order to disambiguate the context, perhaps if we give overtraining to the animals will start incorporating these cues in their reorientation strategies to recover heading, because the geometric errors do not maximize reward. So even though geometry might be a stable landmark, once the animals learn that there is another cue that maximizes reward, maybe they will switch their reorientation strategies. So this is what we have been doing lately. So my student Celia trained animals, giving them more trials per day in the two-chamber task, but now we put more cues in order to make them more salient. And what she found is that after giving the animals 8 days of training on day 1, she tested in the last 4 trials, and she replicated the previous results. The animals show dissociable patterns of digging, showing that the animals are using the geometry to recover the heading. And at the same time, they are recognizing the contexts are different, and that's why they show the dissociable pattern of digging in the two contexts. However, when she continued training the animals, she started seeing that even though they continue using the geometry to some extent, over time, the digging in the correct caps start to increase. You can see that even more on day 3. And the diggings in the geometric corner start to decrease. So she quantified that by combining the diggings in the two contexts. And you can see that on day 1, definitely the digging in the correct and geometric corner are equal. But after day 2 and 3, you can see that the animals dig much more in the correct caps than in all the other locations. So what happens at the neural level? First of all, given that we know the cells will align to geometry, will the alignment continue as the animals start incorporating the non-geometric cues in their strategy? So here, Celia recorded with tetrodes as well as calcium imaging. And here are examples of cells that show perfect geometric alignment with tetrodes. You can see the rotations coincide with 180 degrees. And this is with calcium imaging. We see the same geometric alignment. The way she quantified this is that we had all the pairwise comparisons, and we have all the maps for these pairwise comparisons. So for each pairwise comparison between maps, we leave one trial unchanged, and the other trial is left without change or rotated 90 degrees, 180, or 270. And then we perform pixel-to-pixel cross-correlations of neuronal activity to determine which is the rotation that represents the best match for that comparison. And we use this for all the trials, all the pairwise comparisons for all the animals. And here I summarize the data for tetrodes and calcium imaging. And what is important to note here is that in both cases, 0 and 180 degrees are the rotations that represent the best match of the population, the rotations in the population, and the alignment to geometry persists for the 3 days of training. even after animals go to the correct cup more, the alignment persists, which suggests that the animals are going to the correct cup by combining geometry and landmarks. So they say the correct location is where I see the long wall to my right, but is next to the striped wall, for example. I will provide more evidence for this towards the end. So the next question is, how do the hippocampus codes the context recognition? So there are three different hypotheses here. It is possible that we see I have shown you, we will see alignment, but in between contexts, there are changes in firing rate. Another possibility is that there is alignment, but across contexts, the cells firing in different locations. And within comparisons in the same context, we see the 180-degree rotations. A third possibility is that there is a subpopulation of cells that doesn't align to geometry so strongly, but over time, these cells become aligned to the cues. So this will be the real context cells, because as the animal learns the directional value of the non-geometric cues, they become stable within context, but they remap very strongly across contexts. So the first thing that we tested was the rate remapping across contexts. And for each cell, we had the firing rates in context A and the firing rates in context B. So we created matrices that allow us to compare trials. For example, two trials that were tested in context A. So the upper quadrant will show you within context comparisons in context A. We also could compare differences in firing rate across contexts between context A and context B, which will be represented in this quadrant and this quadrant. And we also could evaluate within context presentations in context B. So the highlighted quadrants will show you the differences in firing rate within context, and the quadrants that are not highlighted, differences in firing rate across context. So you can see on day one, we didn't see differences. On day two, we start to see that across context shows slightly higher differences, but the differences become much stronger on day three. This is quantified in these histograms. So this was very interesting and kind of coincided with the use of the non-geometric cues in the reorientation strategy, but it couldn't explain the results of day one. So then we thought, okay, if the firing rate has information about the context, maybe we can use the withheld trial procedure and use a classifier to see if we can determine whether the withheld trial was recorded in context A or context B. So using this classifier, we found that on day one, we couldn't predict the context, but we could predict the context on day two and much more strongly on context on day three, which coincides with the degree of rate remapping being maximal on day three. As I said, this is very interesting and correlates with the behavior of the animal using the cues, but doesn't explain how the animals perceive the context differently on day one. So the next possibility was that there was some degree of location remapping across contexts. So to evaluate this, first we had to align the maps, because given that the maps align to geometry, if we want to see the degree of similarity across contexts, the first thing that we had to do was to align all the maps. And then we did pixel to pixel cross correlations across context and also within context. So here I am showing you the distributions of the correlations within context, which are the yellow lines, and across context, which are the purple lines. Again, on day one, we didn't see differences, but within context comparisons show higher correlations on day two and day three, indicating that the cells start to remap more. There is more location remapping on day two and day three across context than within context. This was interesting, still couldn't explain what happened on day one. However, I noticed when my students showed me these results that there was a small percentage of cells that show very low correlations below 0.3. When we plotted the distribution of correlations, we saw that the distribution was kind of binomial, with cells that had correlations below 0.3 and the majority of the cells having correlations above 0.3. So I asked her to quantify how many of these unstable, we call them unstable because the correlations are so low, meaning that the similarities within and across contexts are poor. And she found that there were, as we predicted and saw from the cumulative distribution, that these percentages of cells are very low. With the tetro data, it varied from, they range from almost 12% to 20%. But in general, they were very, very low. And this is an example of a cell that is stable after alignment. And this will be an example of a cell that is unstable after alignment. Now, we couldn't do much more analysis on these cells with the tetro data because, as I mentioned, our yield with the tetro data in mice ranges from 15 cells to 40 cells per animal. So you can imagine that if we have only 11% of unstable cells, we are talking about 1 to 2 cells per animal, which doesn't allow us to do much. However, with calcium imaging, as I mentioned, we record from hundreds and hundreds of cells, even though the percentages were low and equivalent, that gave us populations of cells that were of significant size. You know, on day one, we had 211 unstable cells. So the next analysis that I am going to show you is all done with calcium imaging. So the first thing that we did was to look at the within and across stability using calcium imaging. And you can see that the stable cells do not differ across days, which are represented by solid lines within or across. But the unstable cells that by definition, you know, show more stability on day one are more stable within context than across context. So this means that this population of cells allows the animals to distinguish when they are placed in a different context on day one, even before the animals start using the non-geometric cues for their reorientation strategies. Now the differences increase over time. Now, since we see this effect of learning, something else that calcium imaging allows us to do that I didn't mention before is that we can use algorithms to register the cells on day one and then track the same cells over time. Our subpopulations get reduced because we cannot track every single cell we record, and presumably there are new cells that are added to the network. But since we record from many, many cells, still we can track sufficient cells from day one to day 3 with a lot of certainty and look at the effects of learning on the same subpopulations of cells. So the next analysis is only done in the cells that we register and we recorded from day 1 to day 3. So the first question that I had was, okay, do the cells that are unstable show this pattern, the same cells when we register the cells? Do they discriminate across contexts more as they learn about the context? So to do this, we use something called the dot product. So we separated the cells according to whether they were stable or unstable, and whether they were recorded on day 1 or day 3. And then we stack all the maps recorded in context 1 for all the cells and all the maps recorded in context 2. And then we create the vector for each pixel location and multiply that vector with a vector in the exact same location in the other context, which gives us what is known as the dot product. Since we had 600 pixels in our environments, this generated 600 dot products. And what you need to know is that the higher the value of the dot product, the more similar the populations in context 1 and context 2 will be. And the lower the dot product, the more dissimilar the populations will be. So in blue, I am showing you the stable cells on day 1. You can see that on day 3, the same cells become more stable. Interestingly, in red, the unstable cells on day 1 become even more unstable on day 3, I'm sorry. So this means that there is an effect of learning on the same cells. The cells learn to discriminate even more as time goes on. The interesting and more important question was, we see that these cells show this more extreme remapping from day 1 on day 2 and day 3 across context. What happens within context? So are these cells going to stabilize to the local cues and remap very strongly, as I showed you, across context? So to do this, because we don't have two context, what we plotted were the cumulative distributions of the stable and unstable cells within context. And you can see that again, the stable cells become more stable, but the unstable cells become more stable over time, which is this red dotted line. So what this means is that the cells that were stable within context kind of clear the noise and become even more stable, which are the cells that kind of align to the geometry. The cells that were unstable within context over time stabilize within context, become aligned to the non-geometric cues. So these are the true context cells that allow the animals to discriminate the context and also incorporate these cues in their reorientation strategies to maximize reward. So we are wrapping up this to send out for publication. We already finished collecting all the data, but I want to show you some preliminary data that got me super excited. This is totally preliminary, and I have not analyzed everything, but this is so exciting that I cannot avoid showing this to you. Since we saw these effects of learning on the cells that we record, I told my postdoc to do some decoding using the neuronal activity. So you can use the neuronal activity and Bayesian methods to decode the position of the animal. And he came to see me and he said, okay, the decoding works pretty well when the animal is moving, but when the animal stops, I get this prediction error that jumps all over the place. So I will show you the data as I saw it. So you see here, the animal is moving and the X represents the position of the animal and the open circle, the decoding that we do based on the neuronal activity. And you can see that when the animal moves fast, there are a few errors, but when the animal moves fast, we can decode the position pretty well. Now, in a few seconds, the animal will stop there. And now you see that the error jumps all over the place. So I have another movie in a slow motion to show you what the prediction does. So look, now the animal is stopped. Look at what the prediction goes to this location, now this location. It seems like the animal is thinking about all the locations that are important to characterize the shape of this enclosure. In some situations, the prediction even goes to locations that are behind the animal, because at some point someone asked me recently, is this what the animal is looking? It cannot be what the animal is looking because you saw the prediction jump behind the animal. And towards the end, the prediction goes twice to this location, which is the geometric cup, and the animal tends to go there. So I am super excited about this because it seems that the animal is thinking about the shape of the enclosure, and then the prediction jumps twice to the geometric location and the animal ends up going there. So we are doing a lot of analysis on this data, trying to extract meaning from it. We are trying to use a different method for decoding the position of the animal. But I am super, super excited about this. So finally, I want to finish telling you something about where are the geometric inputs coming from and how does the brain control for these inputs? Because I told you the geometry is a more stable cue, and animals use geometry initially to reorient. But in symmetric environments, they do not maximize reward. So the brain must have some kind of mechanism to tell the brain regions that send these inputs, stop sending me these inputs. So to do this, we turn into the retrosplenial cortex, because in human studies, fMRI human studies, the retrosplenial cortex has been shown to be a region important in scene perception. Additionally, the retrosplenial cortex contains cells that respond to location, which we also identify. They have place fields similar to those that you see in the hippocampus. And when we're recording change tasks from retrosplenial cortex, if the animal makes a geometric error, we see that the cells flip 180 degrees. We also see cells that have more than one field. And normally those cells fire on one of the axes that connects the task relevant locations. When we did the geometric alignment analysis, we saw that 0 and 180 degrees align the retrosplenial map. We also identified head direction cells. These cells fire when the animal's head is facing a particular direction on a compass. And you can see that when the animal goes to the correct cup, this head direction cell has a particular direction, but when the animal makes a geometric error, the directionality of the head direction cell flips 180 degrees. And we were also very excited to find multiple head direction cells with multiple directionality, which have been recently discovered by Kate Jeffrey. And we show here that the multiple directionality is controlled by the geometric, the rotational symmetry of the environment. I don't have time to show you, but we also identified egocentric boundary cells that were discovered by Mike Hasselmo very, very recently, that fire related to the position of the animal, where the animal is relative to the border, relative to the borders. So part of what we are doing now is trying to see how the activity of these cells changes as animals are overtrained. Finally, as I said, you know, the geometry leads the animals to make geometric errors. So at the time I started thinking about how the brain tells these geometric regions to stop sending geometric information, Gordon Sheffer, who is a neuroanatomist that uses viral approaches to study connectivity in the brain, came to give a seminar to UTSA, and I was able to talk to him. And he told me about this paper that he was publishing in Nature Neuroscience, where he characterized a projection that originates in area CA1 of the hippocampus and sends axons to layer 1 of retrosplenial cortex. And this is a long-range GABAergic projection, meaning that it is inhibitory. So when he expressed an opsin that activates those cells, which is a method called optogenetics that we have in order to manipulate selective projections, when he activated with a light the terminals of these GABAergic cells, he saw that the neuronal activity in the retrosplenial cortex was inhibited, the activity in the pyramidal cells. So I told him I will use this projection in the context of my reorientation task. So my student Celia use GAT2 mice. So these mice express Cre recombinase under the GAT2 promoter, which is a promoter that is only present in GABAergic cells. And she injected a crimson flex virus. So this means that crimson, which is an opsin that is activated by light, will be expressed only in GABAergic cells. Now, she put a red light, this opsin is shifted by red light. So she put a fiber optic at the level of the retrosplenial cortex in the midline, because these projections end on layer 1. So by putting a light in the midline, she could activate the projections that go to both sides of the brain. So when she turned on the light, these GABAergic cells will release a lot of GABA and will inhibit the cells in the retrosplenial cortex. So she did this experiment on a trial by trial basis. So she trained the animals in Cheng's task, and you can see without the light on, the animals dig in the correct and the geometric error cup. When she turned the light on, the animals went at random. They couldn't use the geometry to reorient, and they were at random. This was early in training. What Celia did, so this suggested that the retrosplenial cortex is important to provide the geometric input to the hippocampus. What Celia did was to continue training the animals until the animals were making 70% correct responses. So the animals can go to the correct response, correct cup, because they say, okay, as I mentioned before, the long wall is to my right, and the correct location is the long wall to the right next to the striped wall in front of me. So when she found that the animals were making about 70% correct responses, in some trials, she turned on the light. And what she found is that now the animals cannot get the geometric input, but the animals still remember that the correct location is next to this feature here. So the animals dig in the correct cup and equally often in the cup next to the reward and next to the feature. So what this is telling us is that the retrosplenial cortex is important for sending geometric inputs. When we silence the retrosplenial cortex early in training, we inhibit the ability of the animal to reorient. When we silence the retrosplenial cortex late in training, when they also combine features, the animal cannot use the geometry and only can use the features and confuse the two cap locations that are close to these features. So we are super excited about this. And in conclusion, what I have told you today is that the hippocampal map aligns to geometry during reorientation, and the alignment serves to predict behavior. Heading retrieval and context recognition are independent cognitive processes that initially rely on geometry and landmarks, respectively. However, as animals learn the directional value of non-geometric features, they incorporate these cues in their reorientation strategies to recover heading. And these processes are dissociable at the hippocampal level. The retrosplenial cortex is necessary for the use of geometric strategies during reorientation. And there is a long-range GABAergic projection that originates in CA1 that serves to inhibit geometric inputs and bias reorientation towards featural strategies. And I will not talk so much about my current directions because I already told them as I was describing the experiments. We are trying to see the effects of overlearning in retrosplenial cortex and understand better the role of this projection by recording at the same time that we stimulate the terminals, and as well as recording from the cell bodies of the GABAergic cells. But in the long-term objectives that I have, I want to evaluate the reorientation representations during aging and neurodegenerative disease. I have a lot of data with aged animals that is super interesting. I didn't have time to show it to you. I want to study the role of post-subiculum, which is a region involved in landmark control, because I believe that's the piece of the puzzle that I am missing. If I am correct, the inputs from the subiculum will be those that allow the animals to use the feature during reorientation. I want to also examine how oscillatory activity changes across the spatial regions during reorientation. So far, we have been using unit activity or calcium imaging. I want to implement the neuropixel technique that will allow us to record from hundreds of cells at a time and from multiple brain regions. And I want to evaluate reorientation in more complex contexts, because we have been training the animals in relatively simple contexts, but normally we get lost in more complex contexts. So once we understand the variables that govern reorientation in simple contexts, I want to make the tasks more complex. And I want to finish by thanking the people that made this possible, my funding sources, collaborators. This project was started by Alex Kinath and Josh Julian, but I want to mention the MVPs of most of the data that I collected today, my current student Celia Gagliardi and my postdoc Mark Normandin. They are absolutely amazing. They are gifted, Celia is a gifted experimentalist, as well as has very strong computational skills. And Mark is a physicist and he is awesome, and he has helped enormously with all the projects that relate to calcium imaging. And also the other members of my lab, Matt Lopez, Nicole Cook, a talented undergraduate who helped with his projects, especially the optogenetic projects, Nishant Punhala. And thank you so much for your attention.
Thank you, Isabel, so much for that talk. The floor is open for questions. Anybody wants to ask a question? You can either type in the chat.
Question.
Sure, go ahead.
Hi, Dr. Muzzio. Thank you so much for the interesting talk. It was so, full of a lot of details. So I was curious about one of the plots that you showed actually on the stability, the number of cells changing within context and how they were changing. So my question is, there were some cells which were stable within context. That means that they were firing at the same spatial location within context. And there were some cells which were unstable, which means that they were firing in different locations, right? So does it mean that those cells were more open to context because they were actually paying attention or open to the surrounding more than the ones that they were concentrated on the same space? Is that why they kind of catch the context more when you change the context?
You are absolutely correct. and you understood this perfectly well. And that's an excellent question. When I show these are the correlations within context, and let me, I don't know, I have to maximize this. I don't want to do something that, it's true that there is still a percentage of cells. We calculate correlations below 0.3 are considered very unstable. Above 0.3, I would say a cell is pretty stable. You never get, I mean, correlations of 0.8, I mean, there has to be almost no noise at all. With correlations of 0.4, I would say already the cells are pretty stable, and above is super, super good. So, but you are right that within the subpopulation of unstable cells, there is still a small percentage of cells that continue to be relatively unstable, even within context. I am not sure if these cells have either poor quality, could also be that the firing rate of these cells is low, because one of the issues in our reorientation paradigm is that we allow the animals to reorient, but we don't leave the animal in the context forever until the animal is totally oriented in the context. So the trials for reorientation are relatively low. So one possibility that could give rise to the cells that are still unstable, even after we do the alignment and we check and we do the registration and so on, could be cells that have a relatively low firing rate because the animal went to that location fewer times than into other locations. So that's something that is possible. I will have to examine more closely what is the nature of this very small percentage of cells. I didn't examine them because this subpopulation of cells is already smaller. So we are talking about very, very few cells. Because when we do the cell registration, we already narrow down the subpopulation of unstable cells. Like, for example, if we start with calcium imaging with 210 unstable cells, after we do the registration, we ended up with groups that had like 90 cells. So you are talking about here probably 10 cells. Not more than 10 cells. So it's very, very small subpopulation of cells. So I'm not 100% what they represent. Could be that particular region where the cell fires has not been sampled as much as the others, or it could be that the cell is just a low firing rate cell. But definitely it's not a significant population because I still see that the majority of the cells have correlation values that are about 0.3, which would indicate stability.
Thank you.
Thank you.
Other questions?
I did have one. Hi, that was a great talk. I did have one question regarding, so in this experiment, we are monitoring individual cells and seeing how they respond to particular geometries and features. Correct. And contexts. But what if the cells that are responding to a particular geometry are not individual cells, but a group of cells or multiple cells in a particular combination? And also that those cells, like a different set of cells will respond to a different geometry feature, but they do share certain cells. So if you were monitoring individual cells, then that one cell would respond to both geometry features. So that would account for some of that error margin that you're having.
Mm-hmm. That's a very good point. I was anticipating that if those subpopulations that kind of are active together in relation to the geometry, I anticipate that those types of representations will be more prominent in the retrosplenial cortex rather than the hippocampus, although I may be wrong, mainly based on the fMRI data that has been collected. So the retrosplenial cortex is a region that is involved in scene perception. And there is a recent paper that suggests that it can kind of integrate scenes, which would be the first step to form a representation of the layout, right? So I think that if there is that type of arrangement that you are talking about, or coordination of neuronal activity, it may happen upstream of the hippocampus. It is possible that it also triggers neuronal activity in the hippocampus. But the literature has shown, for the most part, that, I mean, this may be wrong, that the neuronal activity of hippocampal cells is kind of independent of each other. That doesn't mean that when the cells receive a particular input, that input may not synchronize more the neuronal activity in order to create that perception. And in fact, the decoding experiment is giving us a hint that that could be one possibility. But we will have to do further analysis to really get into that. I am trying to get into that in the retrosplenial cortex now. That's an excellent point.
Thank you.
A great discussion. I think we're slightly over time here. If there are further questions, we'll follow up with you, Isabel. I personally have a few questions, but I know some of the students have to leave because it's, and probably you have other meetings too.
Yes, thank you. I want to thank everybody for joining and giving me this opportunity. And I hope it was not too physiological and you could follow the talk. So thank you so much for your attention.
Thank you.
Bye.
Bye.